In this lecture, we are going to discuss the applications of the hepatitis B virus. Before starting the applications of the hepatitis B virus, we will first of all look at the replication of the genome, either DNA or the RNA, that how DNA or RNA is actually synthesized in the different viruses. So let's get started. First of all, we will look at the replication of the hepatitis B virus, specifically in the hepatocytes. So here is the full structures of the hepatocytes, and we will discuss how hepatitis B virus actually replicate in these hepatocytes. So basically two things actually fall in the genome, DNA or RNA. Okay, so first one is the DNA. DNA can be synthesized from the RNA or the DNA itself. Okay, that's the most important thing. DNA can be synthesized from the RNA or it can be synthesized from the DNA. Okay, so there are basically the two important categories. If the DNA is synthesized from the RNA, then what type of polymerase it requires? It requires the RNA dependent DNA polymerase. Okay, so it required RNA dependent DNA polymerase, RDDP, RNA dependent DNA polymerase. But if the DNA is synthesized from the DNA, then what type of polymerase it required? It required the DNA dependent DNA polymerase, DDDP. So in both of them, you will see the DNA polymerase. So it means that it shows that it will result in the formations of the DNA molecule. Okay. On the other hand, RNA can be synthesized from two different things. RNA can be synthesized from the RNA. Similarly, RNA can be synthesized from the DNA. If the RNA is synthesized from the RNA, then what type of polymerase it required? It required RNA dependent RNA polymerase. Okay, that is RNA polymerase. Next, if the RNA is synthesized from the DNA, then it will require the DNA dependent RNA polymerase. So RNA polymerase will be common in them. Okay, but in the replications of the hepatitis B virus, we will look at the DNA replication, because hepatitis B virus is actually DNA virus. Okay, so it will require the DNA polymerase, and what that DNA polymerase has ability to synthesize the DNA of the hepatitis B virus either from the RNA or from the DNA. Important thing which you have to remember, synthesis of the DNA from the RNA happens in the presence of the RNA dependent DNA polymerase, which is also known as reverse transcriptase enzyme. Okay, that's the most important thing. Okay, so first of all, if we look at the structure of the hepatitis B virus, so outermost is the, here is the outermost envelope of the hepatitis B virus. Here is the envelope of the hepatitis B virus. Next, you will see the capsid of the hepatitis B virus. That will be the icosahedral in shape, means 20-sided shape. Okay, and inside the capsid, okay, here is basically the capsid that is actually made up of protein, and inside the capsid you will see the presence of the genome, and that will be the partial DNA. Okay, what does it mean? It means that DNA is not complete. Okay, one strand is complete, that will be the negative, and second strand is incomplete. Okay, that strands will be the positive. So basically DNA has two strands. Here is the first one and here is the second one. So one strand is positive polarity and second strand is negative. But in the hepatitis B virus, you will see the one strand is complete while the other strand is incomplete. So that's why we call the hepatitis B virus has incomplete DNA. Inside the capsid, you will also see the presence of the DNA polymerase. This one, DNA polymerase. And this DNA polymerase has ability to synthesize the DNA from either RNA or DNA. Okay. Now, this hepatitis B virus will infect or attack the hepatocytes. Why? Its receptor. Okay. So on the hepatocytes, you will see the presence of the receptor, a specific receptor. So that's why hepatitis B virus is specifically attack on the hepatocytes due to the specific receptors present on the hepatocyte. So that receptor is actually the sodium taurocholate. Sodium taurocholate. These are the receptors that are actually present on the hepatocytes. Okay. When this hepatitis B virus affect the hepatocytes, okay, then during the attack, you will see the envelope will remain attached to the cell membrane of the hepatocytes, while only the capsid will release into the cytoplasm of the hepatocytes. Okay, that's the most important thing. Here is the complete strand. Inside you will see the incomplete strands, and you will also see the presence of the DNA polymerase. Okay. Now after that, that capsid will move towards the nucleus of the hepatocytes. Okay. So here is basically the nucleus of the hepatocyte. While this process will happens through the microtubules actually present in the hepatocytes. Microtubules, that is actually the skeleton of the cell. Okay. That capsid will move towards the nucleus. Okay. And after that, that capsid will attach to the nuclear part of the nucleus. Okay. Here is the complete strand, and you will also see the incomplete strand, and you will also see the presence of the DNA polymerase. So here are basically the important things: DNA polymerase, and here is basically the negative strands, and here is basically the positive strand. Okay. When the capsid attached to the pores of the nucleus, okay, here are basically the different pores of the nucleus, then only after that you will see only the DNA will be entered into the nucleus. Okay. So inside you will see here is the negative strands of the DNA. Okay. And here is the positive strands of the DNA. Okay. And here is the DNA polymerase enzyme. Okay. Now, as we have discussed, that DNA polymerase can synthesize the DNA from the DNA. So it is actually a DNA structure. So, but it is actually incomplete. So this DNA polymerase will complete this DNA, and ultimately you will see the two strands. One is negative polarity and other is positive polarity. But in that case, you will see the complete strands of the negative and the positive. Now this DNA is actually complete. That is actually called the cccDNA, covalently closed circular DNA. Now that DNA is actually complete. Okay. Next step, you will see two phases. First is that it can enter into the replication phase. Okay, that's the most important thing. And it can also go towards or attach to the genetic material of the hepatocyte itself. Okay, that's the most important thing. If I say here is the DNA of the host cell, host cells means DNA of the hepatocytes. Okay, now this DNA can attach to the DNA of the host, means DNA of the hepatocyte. That form actually happens in case of chronic carriers, chronic hepatitis. That's the most important thing, that ultimately go towards the hepatocellular carcinoma. Okay, next you will see from the DNA, you will see the synthesis of the messenger RNA. As the normal process happens, from the DNA, you will see the synthesis of the messenger RNA. From the messenger RNA, you will see synthesis of the different proteins. So similar process will happen in that way. Okay. From the negative strands of the cccDNA, you will see the formations of the positive polarity messenger RNA. That's the most important thing, and that process is actually called the transcription. So you will see the synthesis of the messenger RNA. In that process, you will see what type of polymerase it required. It required the DNA dependent RNA polymerase. Okay, it required DNA dependent RNA polymerase, and that polymerase will be host dependent. Okay, means that DNA is actually comes from the host. So from the negative strand, you will see the formation of the positive polarity messenger RNA. Okay, that will forms the two things. Okay, it will move outward and perform two most important things. Okay, so here is the endoplasmic reticulum. Okay, so endoplasmic reticulum has two types, smooth and the rough. In the smooth, there is no ribosome. Okay, and on the rough endoplasmic reticulum, you will see the presence of the different ribosome. As we know that the translation or the formations of the protein process happens on the ribosome. Okay, that messenger RNA will go towards the ribosome. Okay, and in the ribosome, you will see the synthesis of the proteins. Okay. Now, in that case, you will see the synthesis of the different proteins. Okay. Now, these proteins will get the important antigen, that will be the hepatitis B surface antigen. Okay, that's the most important thing, and go outward from the rough endoplasmic reticulum. Okay, here are basically the different proteins that will ultimately used in the formations of the capsid. Okay, and next, this messenger RNA, here is actually the messenger RNA, and the polarity of the messenger RNA is actually positive. Now, from the positive polarity messenger RNA, you will see the synthesis of the negative polarity DNA. Negative polarity DNA. Okay, now you see the synthesis of the DNA from the RNA, and in that case you will see the RNA dependent DNA polymerase, or you can say reverse transcriptase. Okay, and again, now this negative strands of the DNA will act as a template, and it will result in the formations of the positive polarity DNA. Here is the negative DNA, and it will act as a template, and it will result in the formations of the positive polarity DNA. Here is basically the two strands of the DNA, but that DNA will be incomplete. Okay, now both important two components of the virus, that is the genomic material. Here is the genomic material, here is the proteins that is actually used in the capsid. Okay, these two actually move towards the cell membrane. Okay, from the cell membrane, in that case you will see the budding of the hepatitis B virus. Okay, in that case you will see the capsid. Okay, and you will see the two incomplete strands of the hepatitis B virus, and you will also see the presence of the polymerase. Okay, that budding happens through this way. That's the most important thing. That is the whole process that actually happened into the hepatocytes. Okay, that hepatitis virus will bud from the hepatocytes and carry the important hepatitis B surface antigen. But important thing, that antigen will now be present at the membranes of the envelope. That will be the hepatitis B surface antigen, most important component. Okay, but important thing, that whole hepatocytes will be destroyed by our immune system. How? Okay, that is the normal replication process of the hepatitis B virus. Okay, but important thing which you have to remember, that the hepatocytes have major histocompatibility complex, that is the MHC. Actually, MHC present the antigens of the hepatocytes on the surface. If I say major histocompatibility complex will represent the antigen outside the hepatocytes, and it will result that lot of different cytotoxic T cell will detect this antigen and will destroy the whole hepatocyte. So it means that our immune system will destroy the hepatocytes, and it will result in the damage of the liver that can go towards the cirrhosis and ultimately can go towards the hepatocellular carcinoma. That's the important thing. Virus directly not affect the liver, but our immune system will affect the liver due to the presentations of the hepatitis B antigen through the major histocompatibility complex that are actually present on the surface of the hepatocytes. So this is all about the replications of the hepatitis B virus, that is also known as the DNA particle. If you still have any question, you may ask in the comment section. Thank you so much.